This is lecture 19, and we'll be going over some immunology procedures that are more common in the laboratory that uses precipitation. First, let's review some agglutination testing. So if you'll fill out this chart or write it down on a piece of paper, we'll go over the answers in the next slide. For direct agglutination, the known can be either the antibody or antigen, and the unknown can either be the antibody or antigen, and we do not have a carrier in direct agglutination. Passive agglutination, our known is the antigen and our unknown is the antibody, and typically we will have a carrier like a latex bead, etc. Now remember, if we're dealing with passive hemagglutination, the carrier would be an RBC. We have reverse passive agglutination, which we have our known as the antibody and our unknown antigen, and we will have a carrier. And again, we could have reverse passive hemagglutination as well, where the RBC is the carrier. In co-agglutination, we have our known antibody and unknown antigen, and we have bacteria that is the carrier. Remember, typically this is usually Staph aureus. We have agglutination inhibition. We have our known antibody and unknown antigen. And typically, yes, we do have a carrier with our known antigen. Now remember about agglutination inhibition, that positive clumping means a negative result. In this lecture, we'll be talking about rocket immunoelectrophoresis and immunofixation electrophoresis, and we'll briefly touch on the Western blot. The first two electrophoretic techniques we're going to talk about are rocket immunoelectrophoresis and immunofixation electrophoresis, or IFE. Now these techniques use electricity and diffusion. That way we could get a faster result and more of a sharper image in our results. Rocket immunoelectrophoresis is typically used to quantitate antibodies present in body fluids. So we'd have our known antibody and unknown antigen. So basically the known antibody is distributed in the gel and antigen is placed in the wells. We will use electrophoresis to help facilitate the migration, and then precipitation will occur in a rocket-shaped pattern when these antigen and antibody complexes form. And in the next slide we'll see an example of this rocket shape pattern. In rocket immunoelectrophoresis, it is very pertinent to have three different controls. So in these controls, we have different amounts of antigen in each control. So this would be directly proportional to the amount of antigen we put into the well. So we'll have a low antigen content, medium antigen content, and a high antigen content. Then we will run our patients as well, and then we'll compare our patients to our controls, and this is how we get our results. So the height of the rocket is directly proportional to the amount of antigen in a particular sample. So in our controls we have a low, medium, and high, and in our patient samples we're going to determine the amount of antigen from our controls compared to our patient samples. And this is rocket immunoelectrophoresis. Before I get into immunofixation electrophoresis, or IFE, I want to be clear that IFE is not the same thing as immunoelectrophoresis. So in immunofixation electrophoresis, like we're about to talk about, we're going to be putting the stain directly onto the gel. In immunoelectrophoresis, we put the gel in a trough and stain it that way. So that's the main difference between IFE and immunoelectrophoresis. So the IFE, we can use this to detect antibodies or antigens through agarose or cellulose acetate, the gel, basically. So it has basically two stages. So we have our test material with our unknown antigen. We'll send that through the electrophoresis to help separate these antigens. Then we'll add specific antiserum to the gel, wash it to remove any non-precipitated proteins, and then we'll stain the precipitated proteins. Alright, and this is what gives us our marks across the electrophoresis gel. We use the IFE method quite a lot, especially here at UAMS. We have a multiple myeloma center, so we're doing a whole bunch of IFEs for in order to help diagnose patients with multiple myeloma. So people with multiple myeloma have excess antibodies made by malignant plasma cells, and typically we can detect these excess antibodies either through the patient's serum or urine, and we can electrophorese these out to help separate these antibodies and detect what is going on, what is that excess antibody that they are producing, and kind of go from there. When we're talking about multiple myeloma patients, we usually try to detect the patient antibody that is in excess. So we consider this in the IFE test to be the antigen. So we could get serum or urine from our patient, we'll add it to the lanes, we'll electrophorese it out, and then we'll add a specific anti-human antibody or a reagent. Okay, this will infuse cellulose acetate strips are placed on the separated proteins, will allow the antibody to diffuse into the gel, resulting in the antigen and antibody complexes. Alright, so we have our antigen from our patient sample, and then we have our antibodies from our reagent. We'll combine those together, allow those to diffuse and form complexes. Then we'll stain the strips and examine for patterns. And in the next slide we'll see an example of a IFE pattern. So on the very far left, this is our control. So we have our albumin, our alpha 1, alpha 2, beta 1, beta 2, M band, and then our wells are the clear ones at the bottoms. So remember when we were talking about serum protein electrophoresis, or SPEs, this is the same thing. It's just not in that little curve in a graph. It's flat. So we have our big albumin spike, we have our alpha 1, alpha 2, beta 1, beta 2, and our M band, which is our gamma band. So when we electrophoresis our patient out, we typically will test for IgG, IgA, and IgM. And we'll also test for our light chains, kappa and lambda. Alright, so in this particular example, we have a anti-IgG and anti-kappa. So we have a monoclonal IgG and kappa light chain disease. And this is more of the same thing from the previous slide, but an actual gel that we performed. In this example, we have a monoclonal IgM with kappa light chain disease, because we can see in lanes 4 and 5 we have that dark band, and that tells us that we have a monoclonal IgM and kappa light chain disease. I'm going to briefly talk about the Western blot. The next few slides will kind of go over the procedure, etc. Some labs still use the Western blot, some do not. It is becoming pretty much non-existent. But here I am going to give a very brief overview and breakdown of the Western blot. So we used to use this as a confirmatory test for HIV. In some labs we still do. So the Western blot actually helps determine the presence of antibodies to complex antigens such as HIV, Lyme disease, and syphilis in some cases. Usually it will have like a two-tiered testing involved. We'll have an EIA test and then an immunoblot, which is the Western blot. And if both of those are positive, then it is a confirmed positive. In no way is this procedure that I have on this slide the full process of a Western blot. It is super labor intensive. This is just the general outline of the Western blot. So we have our known antigens, which undergoes electrophoresis to separate the molecules. We'll blot this onto nitrocellulose paper, alright, aka sandwich. With the gel, we will put it through an electrical field to pull the bands out of the gel to absorb into the paper. We'll add the patient's serum, we'll incubate, and rinse, stain, and then we'll look for the bands. That is the most simplistic way of the Western blot, but there are the procedure itself is much more detailed than this. Typically, the Western blot is used in polyacrylamide gel, so this gel usually stands up vertical instead of horizontal like in normal electrophoresis, and so this would the direction of migration would be down instead of across. We then remove the gel and then blot it on cellulose paper. Alright, and this is why one of the reasons why it's called a blotting test, because we're blotting the cellulose paper with the gel. And then we would add the patient sample, incubate, stain, and then look for bands. And this is just a carryover from the previous slide. So we have our nitrocellulose strip where we blotted it. Then we're going to incubate our serum from our patient, rinse, then add enzyme labeled antibodies, incubate and rinse, add our substrate, and then look for the color development areas on the nitrocellulose paper to try to identify the antigens that may or may not be present. So when we're doing the Western blot, we're looking for specific bands in the HIV virus. And typically we're looking for 3 specific bands. We're looking for the P24, the GP41, and the GP120/GP160. Alright. So if 2 out of 3 bands of these bands are present, then we would consider this to be a positive reaction. So this person would be positive for HIV. And here's an example of a Western blot. So we have our positive and negative controls, and you can see that our patient number 1 does have the P41 and the P24 marker, so therefore we would consider this to be a positive reaction. So patient number 1 does have HIV, patient number 2 and 3 are negative. So what kind of errors can we have when we're doing electrophoresis? Obviously, the rookie mistake would be to apply the current in wrong direction. I am one of those rookies that I have made that mistake before when I was in school here at UAMS. I did do that, and I watched the antigens run right off into the liquid suspension. So yes, so I have done that. That's because I was not paying attention, but that is a great learning curve. I mean, you will only do that once. We could also have the incorrect pH or separation time. The current could be too strong or too weak, and the ratio of antigen-antibody proportions could be misaligned. In the next few slides, I'm going to be showing you pictures and questions. I'll read the question, then kind of give you a minute to answer, and then we'll move on to the next slide. So what test is this diagram showing a picture of? This would be rocket immunoelectrophoresis. All right, next. What test is this showing? This would be IFE, or immunofixation electrophoresis. What type of test is this? This would be agglutination. And what test is this? This would be nephelometry. Remember, nephelometry is the measurement of side scatter, the light side scatter. Turbidimetry is direct, so the light source would be directly in front of the detector. And what precipitation test is this? This is the Ochterlony double diffusion test. What is this labor-intensive test called? This would be considered the Western blot. Okay, so here are some true or false questions. If you'll answer these, we'll go over them in the next slide. So the first one, turbidimetry measures light scattered at a particular angle from the incident beam. That is false. So nephelometry measures the light scattered at a particular angle. Turbidimetry measures the direct light. Okay, number 2, in the Ochterlony method, both antigen and antibody diffuse. This is true. IFE involves separating unknown antigens with diffusion, adding known antibodies to the gel, and staining for antigen-antibody combinations. This is false. IFE does not involve diffusion. It uses electrical current. Western blot is an adaptation of IFE, but antigens are transferred to a membrane by blotting. This is true. Here's a matching review. If you'll pick out what goes with what, I will give you the answers on the next slide. This uses current to separate unknown antigens, add known antibody, stain, and read. That would be IFE. Measures light scattered at an angle. That would be nephelometry. Known antibody in gel, unknown antigen in well, and add electrical current. That would be rocket ID. Use current to separate unknown antigen, transfer to membrane, add known antibody, stain, and read. That would be Western blot. Measures light scatter in a direct line. That would be turbidimetry. And double diffusion would be Ochterlony. Are we done yet? Why, yes we are. We are finished with agglutination precipitation procedures. We will be talking about nucleic acid probes and PCR in the next 1 or 2 lectures.